Hey there, science fans. Today, we're diving into the world of animal research in India. It's a fascinating area with big responsibilities. We need to make sure our furry, scaly, and feathered friends are treated right, even when we're learning from them. That's where the CPCSEA comes in. CPCSEA stands for Committee for the Purpose of Control and Supervision of Experiments on Animals. Big name, right? But it's for an important job. This committee was set up in India to make sure that any experiments involving animals are ethical and humane. Think of them as the animal kingdom's lawyer in the lab. The CPCSEA was created because as scientists we have a duty to care for the animals we work with. Animals are living beings just like us. They feel pain, they get scared, and they deserve our respect. The CPCSEA helps us remember that. They do this by creating guidelines that scientists and researchers have to follow. These guidelines cover everything from how to house the animals to how to keep their pain to a minimum. It's all about making sure the animals are treated as humanely as possible. So, what exactly are these guidelines? Let's break it down. One of the most important things is getting permission. Before any scientists can even think about working with animals, they need to get the green light from something called the Institutional Animal Ethics Committee, or IAEC. This committee reviews the proposed experiment to make sure it's really necessary and that there's no other way to do the research. Next up, housing. Imagine you're a lab rat. You wouldn't want to be stuck in a tiny, uncomfortable cage, right? The CPCSEA says animals need enough space to move around and the place has to be clean and safe. They even need things to keep them entertained, like toys for mice or branches for birds. Now let's talk about pain. The CPCSEA is very clear. If an experiment might hurt an animal, scientists have to do everything they can to minimize that pain. This might mean using anesthesia or pain relief, just like a doctor would use for a human patient. And last but not least, documentation is key. The CPCSEA wants to know everything about the animals involved in research, from where they came from to what happened during the experiment. This helps make sure everything is done by the book. Okay, so we've covered how to treat the animals, but what about the science itself? That's where good laboratory practices, or GLP, come in. Think of GLP as a set of rules for doing science the right way. It's like having a recipe for your experiment, making sure every ingredient is measured perfectly and every step is followed precisely. Why is GLP so important? Well, it boils down to trust. When scientists follow GLP, it means other scientists can trust their results. They know the experiment was done carefully and accurately. This is crucial because science builds on itself. New discoveries depend on the reliability of old ones. GLP covers every aspect of a scientific study, from planning the experiment to reporting the findings. It's like a roadmap that guides researchers through the entire process, making sure they don't miss a turn or take a shortcut. Now you might be thinking, this sounds complicated. And you know what? You're right. GLP does involve a lot of details and paperwork, but trust me, it's worth it because when it comes to science, accuracy and reliability are everything. Section 4, Principles of Good Laboratory Practices. Let's break down the key principles of GLP so you can see how it works in action. First up, we have planning. Before scientists even put on their lab coats, they need a solid plan. This means figuring out exactly what they're trying to test, what materials they'll need, and how they'll collect and analyze their data. Next comes execution. This is where the experiment actually happens. GLP requires scientists to follow their plan carefully, documenting every step of the way. They also need to make sure their equipment is calibrated correctly and that they're using the right techniques. Monitoring is another crucial aspect of GLP. It's like having a quality control inspector for your experiment. Scientists need to constantly check their work, looking for any errors or inconsistencies. They also need to keep track of their materials, making sure they're stored properly and used before their expiration dates. Of course, all that careful work would be useless without proper documentation. GLP requires scientists to keep detailed records of everything they do, from the initial planning stages to the final results. This documentation serves as a permanent record of the experiment, allowing other scientists to review the work and verify its accuracy. Finally, we have reporting. Once the experiment is complete, scientists need to share their findings with the world. GLP guidelines dictate how these reports should be structured, ensuring they're clear, concise, and complete. This allows other scientists to understand the research and build upon it. Section 5, Bioassays and Introduction. Now let's shift gears and talk about a powerful tool in the scientist's toolkit, the bioassay. You might be thinking, what? Don't worry, it's not as complicated as it sounds. A bioassay is simply a test that uses living organisms or their components to measure the effects of a substance. Imagine you're trying to figure out if a new cleaning product is safe for humans. You could test it on cells in a petri dish to see if it's toxic. Or you could test it on bacteria to see if it stops them from growing. These are both examples of bioassays. Bioassays are like detectives in the lab, helping us understand how substances interact with living things. They're used in a wide range of fields, from medicine to environmental science to agriculture. One of the most important uses of bioassays is in drug development. Before a new drug can be given to humans, it needs to be tested for safety and effectiveness. Bioassays allow scientists to do this in a controlled environment, using cells or animals as stand-ins for human patients. Section 6, Types and Applications of Bioassays. Bioassays come in all shapes and sizes, but they can be broadly classified into 3 main types: in vivo, in vitro, and ex vivo. Let's break down what each of these means. In vivo, bioassays are the ones that use living organisms like mice or rats. This type of bioassay gives us the most realistic picture of how a substance will affect a whole organism. However, it's also the most complex and expensive type of bioassay. In vitro bioassays, on the other hand, are done in a controlled environment outside of a living organism. This usually means using cells grown in a petri dish. In vitro bioassays are simpler and cheaper than in vivo assays, but they don't always give us a complete picture of how a substance will behave in a living organism. Finally, we have ex vivo bioassays, which fall somewhere in between in vivo and in vitro. These assays use tissues or organs that have been removed from a living organism. For example, scientists might study how a new drug affects a piece of heart tissue that's been kept alive in a lab. Each type of bioassay has its own advantages and disadvantages, so scientists choose the best approach based on the specific question they're trying to answer. But regardless of the type, bioassays are essential tools that help us understand the world around us and develop new technologies to improve our lives.